The membrane proteins give the membrane their characteristics and their functions, and I'm going to now explain you 5 different types of membrane proteins and what they do. You already know that a membrane controls what goes in and what goes out of a cell, or what goes in and what goes out of a cell organelle. However, there are membrane proteins which are responsible for other tasks than just the transport of substances. And I'm going to give you now an overview over these examples. So, first of all, one of the examples is... no, that was the wrong one. This one is where I want us to start off with. The first example is of a membrane protein is our receptors, hormone receptors. And in this case, an insulin receptor which can be found on cells. And instead of drawing you now the full phospholipid bilayer membrane, I'm just going to draw over here a double line. No, this is not a double line. Well, it is a double line, but it's supposed to represent my phospholipid bilayer. The phospholipids are in here, but I'm not going to draw them right now, okay, because that's not relevant. And in here, I'm going to draw the protein, namely the hormone receptor. And it would be nice to give it a certain color. It's not possible for me to fill this in with one solid color, so I have to do it like this. And this one is now my hormone receptor. I'm not going to label it as hormone receptor because I already mentioned it over here in the title. And there is a certain three-dimensional shape to this receptor, and over here I have my hormone insulin. And this hormone insulin is now able to bind to this receptor in here. It's able to dock basically onto it. And this now triggers inside the cell here a response. I'm just going to draw it like this, like a little flash. This is a signal, a chemical signal to the nucleus. Basically telling the cell to respond. And that's of course outside the cell. And this is inside the cell in the cytoplasm. And that was already example number 1. So you have receptor proteins which are able to receive hormones. Insulin in this case is the hormone transported over the blood to the target cells, and then the insulin or the hormone binds against the receptor and therefore triggers a chemical reaction, sending a chemical signal to the nucleus. So, next example. The next example of a membrane protein is calidrin, which is responsible for cell-to-cell adhesion. It allows cells to stick together. That's important, otherwise multicellular organisms are going to fall apart. And what we have over here is we have again our protein, calidrin, and it's actually much larger than this. It goes out like that. That's part of the protein. I'm going to color this in here. You probably will do a better job in your notebook. So, and that's pretty much it. This is how the protein looks like. Now, you wonder, how is this important or able to cause cells to stick together? Because over here at the very tip, it's able to attach to another protein of the adjacent cell, and it's basically this sticks the cells together. So if I have another cell here at the top with a membrane, it also has a protein, and it's able these proteins are able to interact with each other, and the cells stick together. And this was already example number 2. There is not a lot more to say here. Maybe you might want to add or ask yourself, is this a peripheral protein or an intrinsic protein? Well, of course, it's an intrinsic protein because it goes all the way across the membrane over here. Next example. Electron transport, cytochrome C is the example. This is important especially for the higher level people. When talking about cell respiration, you have an electron transport chain. And I'm going to really simplify this drawing over here. What we have is we have a protein in here. And this protein is able to accept and also to pass on electrons. So what we have is we have an electron E minus being carried by a peripheral protein called cytochrome C to this cytochrome oxidase over here. So I'm going to draw the cytochrome C over here. That's supposed to be the cytochrome C, and here I think I have to label this. This is the cytochrome C, and this is the cytochrome oxidase. And the electron is in the cytochrome C, and it passes on the electron. Okay, I cannot draw the arrow right now. It's supposed to be an arrow to the cytochrome oxidase, which is an integral protein, and it's an enzyme at the same time, and it is able to accept the electrons. So that is basically the example of electron transport. So I will move on to the next example. And the next example is channels for passive transport. In our case, for example, sodium channels. And what I have over here is I have over here my transmembrane protein, integral protein. In the middle over here, I have a channel. I cannot erase lines very easily over here in this program, so you might want to do that. So I'm coloring it in simply to simplify the drawing a little bit. And we have Na plus sodium, and sodium is able to diffuse across the membrane through the channel protein. And because the diffusion is aided by the channel protein, this is the process called facilitated diffusion. Okay, and this was already the example. It's a passive transport, means there is no energy required. So, and we also have the next example is the example for active transport. In our case, we have a calcium pump. There's also a sodium-potassium pump, for example. And what we have over here is we also have a protein which goes all the way through. But this protein is now a little bit special because it's able to pump the substances from an area of low concentration to high concentration. So what we have is we have calcium, Ca++, but we have over here a low concentration. And down here we already have a lot of calcium. So if we want to move the calcium into a region where we already have a lot, then this requires energy. Again, I'm going to color this in a little bit over here. Maybe I should draw some kind of a channel over here as well. And then I'm going to give you a different example here to actually show how the protein flips over. So, but right, it looks like a channel protein, but actually it's not, because over here we have ATP, and this ATP carries energy and it's hydrolyzed. It's broken down to ADP plus PI. Energy is released in this process, and the calcium is pumped from an area of low concentration to high concentration. Now, it looks like a channel protein, so I'm going to make a different drawing over here. What happens is, one way of imagining this is that you have a protein that looks like this, and here again I have the problem that I'm not able to erase the line over here, and the calcium over here goes in here, and then the protein flips over like this. This requires energy, and then the calcium is able to leave on the other side. So that is one way of imagining how a transport pump works. But in this case, of course, also we need ATP. Okay, just the same thing over here. So that is basically an example for a pump for active transport. And now there is a sixth example for which I just remembered that I should actually include that. I go back over here. If you remember this one over here, I mentioned briefly this is an enzyme. So what I'm going to do is the following. I'm just going to add another one over here, enzymes, as the sixth example. Membrane bound, or membrane immobilized is better. Membrane immobilized enzymes. And what this means is the following. Just add this as a sixth example. It's important also in digestion. Again, also higher level people have to know this. What we have is we have a transmembrane protein over here, and this one has a specific three-dimensional shape. I'm just going to do it like this. I'm going to draw this schematically, and this is supposed to be our enzyme. It's the enzyme. And what we have is we have two substrates, and these substrates, one substrate looks like this, and the other substrate looks like this. And you can probably already see that they kind of fit in here. Okay, so what they do is they bind against these enzymes. So that's substrate 1, this is substrate 2. They bind against the enzyme. I'm going to draw it like this. And what happens is as they bind, they react, the bond is broken, and what we have is we have a finished product, which is P. This is the product which is formed. Okay, so they bond together over the functional groups between substrate 1 and substrate 2, and the enzyme in the process is not modified, it's not changed, and at the end the enzyme will look just like this again, and is able to react the next substrates. So this is basically that's basically it. This cytochrome oxidase over here also works like an enzyme. I mentioned this, but I did not specifically explain how this works because the example here is actually for electron transports, which is this cytochrome C over here. So this is the reason also why I included the enzyme example over here on this form. Okay, so these were several examples of the importance of membrane proteins and what they're able to do.